Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we have been discussing about histone modifications. We are almost done with the histone methylation part. In last lecture, we discussed about all of the important methylation marks that are out there. So we have covered all of them, all of the important ones in the last lecture. But we have only discussed about the lysine methylation. Okay, so we are yet to discuss about arginine methylation marks. So we discussed about H3K9 methylation, H3K27 methylation, H3K20, H3K4 methylation, and H3K36 methylation. The enzymes which are driving this reaction. Okay, so in today's lecture we are going to discuss about those enzymes in detail. We just got to know in brief in last lecture that which enzyme adds which methylation mark, but today we will also know about the other enzymes as well. So the histone, if when they are methylated, okay, so assume this it's the H3 histone, and on the ninth lysine residue it is being methylated. Okay, so this reaction is driven by, from the methylation reaction is driven by the enzymes known as HKMTs, histone methyltransferases. Okay, if they are adding this on the arginine residue, if the methylation mark has been added on the arginine residue, those enzymes are known as protein arginine methyltransferases. Okay, we have already discussed about that, but this reaction is not irreversible. Rather, it is reversible reaction, and the reaction in other direction is driven by the histone demethylases. Okay, this is just the way it happens in the DNA methylation. The DNA methylation is driven by DNMTs, DNA methyltransferases, while the DNA demethylation is driven by the DNA demethylases. Okay, the TET protein, the TET family. Okay, 1011 translocation enzymes. So this was about DNA methylation, but it is the same way for the histone methylation part, but the enzymes involved has different characteristics. Okay, so let's first discuss about histone methyltransferases, HKMTs. Okay, histone lysine methyltransferases. We will discuss about those. So there are two categories of those enzymes based on the domain that they have. So the enzymes which has set domain are categorized in one group, while the enzymes which do not have set domain, so non-set domain containing enzymes. So there are two categories. Okay, let's take a look at the examples of set domain having HKMTs. So we have already discussed about all of those enzymes and which of the methylation marks they add, but let's list them down once. So the enzymes are SUV39H1. The other one is SET1. Okay, the other one which I briefly mentioned was G9A. Okay, there are more like SET1, SET2. Okay, the MLL proteins, the MLL enzyme, and the EZH. Okay, EZH1 and 2. Okay, so those are the enzymes which has set domain. So the so the SUV39H1, SETDB1, and G9A, all of those three enzymes do the H3K9 methylation. Okay, the SET1 enzyme does the H3K4 methylation. Okay, the SET2 or say SETD2 that we discussed in last class, okay, so there was a mistake in the last class. I referred it as the SETD1, but it's actually SETD2. Okay, so it does the H3K36 methylation. Both of those are active marks, well, this is a repressive mark. Okay, the MLL proteins, they also add the H3K4 methylation mark. Okay, while EZH1 and EZH2, those add the H3K27 methylation mark, which is associated into PRC2 complex silencing. Okay, the silencing which is mediated by PRC2 complex. So all of those enzymes have the set domain. That is why they are categorized in set domain containing HKMTs. while the other enzymes which do not have set domain, there's only one example which is worth noting here, and it is DOT1L. Okay, so this is a very interesting example. Why? Because it just not it only doesn't, like, the characteristic that it do not have set domain, but alongside that it is very important because it does demethylation of 79th lysine residue. Okay, it does it adds the H3K29 methylation mark 79. Okay, it adds the H3K79 methylation mark. And why it is so distinct? Because all of this methylation mark, all of those lysine residues, the ninth one, the fourth, the 36th, the K4, K20, the 27th lysine residue. Okay, I forgot one. Okay, I forgot one enzyme which is SUV20H1 or SUV20H2. Those enzymes, those add the H3K20 methylation mark. Okay, so what was I discussing? So the all of those lysine residue, the 9th, 4th, 36th, 27th, and 20, all of those lysine residue, they lie in the histone tails. Okay, so they lie on the histone tails, but the 79th lysine residue, the K79, this is included into histone core. Okay, all of those are on histone tails, the N-terminal long polypeptide stretch. Okay, N-terminal tails. I hope you remember that when we were discussing in the introduction video of the histone methylation, we discussed that and even in the histone modifications lecture, we discussed that almost all of the histone modifications are done on the histone tails, but there are certain modifications which are done on the histone core as well, and this is the example of that one. Okay, so this DOT1L specific methylation is the example of the histone core modification. Okay, so this is a very interesting example. That is why I told you that you have to remember it. So DOT1L enzyme, this the characteristics of the DOT1L enzyme is that it modifies histone core. And even remember the residue, it is the 79th residue. So that's it. Take it as a summary of all of the enzymes. Okay, so that's it about HKMTs. The important fact that you have to remember is that all of the HKMTs have set domain. Only one enzyme, only one category is that which do not have set domain is DOT1L, and it methylates the 79th residue. Okay, so now let's discuss about the histone demethylases. Okay, so just like the DNA methylation, the reaction in opposite direction, this one, the from H3K9 methylation to H3K9, this was thought to be this was supposed to be only one direction reaction. Like, they thought that this is irreversible reaction, so you can't demethylate the histone. Just like in the DNA demethylation, the researchers got to know that DNA can be demethylated when they discovered about 5-hydroxymethyl-cytosine. Okay, so in that way, when the scientists discovered an enzyme known as LSD1, they got to know that the histone demethylation is possible. So in 2004, the LSD1 enzyme was discovered. So in the histone demethylases, we are going to discuss about LSD1. Okay, so this LSD1 is lysine specific demethylase. Okay, so it only demethylases the lysine residue methylation. Okay, so this LSD1 has the amino oxidase activity. Okay, so which is FAD dependent. Okay, means this enzyme needs FAD as the cofactor. So this is the cofactor of the enzymatic reaction. Okay, So there's one interesting fact about LSD enzyme, that the activity of LSD1 is context dependent. Okay, how? Like it depends on the substrate. It is substrate dependent, unlike other enzymes, like unlike the all of the histone methyltransferases we discussed that, above. All of those has certain, like they have certain substrate, like they have the K9 or K4 K36, those as the substrate, and they add the methyl group. But LSD1, it varies. If the substrate varies, the activity also varies. So the activity of LSD1 is context dependent. Like it depends on with which receptor it is acting, or with which complex it is acting. Depending on that, the substrate also changes. For example, when this LSD1, this acts with the androgen receptors, okay, androgen receptors, it demethylates, okay, so it demethylates H3K9 methylation mark. So it removes this methylation mark. Okay. So you remember that the H3K9 methylation mark, this is a repressive mark. Okay. So due to removal of this repressive mark, now there will be the gene activation. So this will lead to gene activation. Okay, removal of the K9 methylation will lead to gene activation. That is why LSD1 here is acting as a activator. Instead of acting as a repressor, it is acting as activator. Okay, it is acting as the activator. So this is important one. But when it acts with the LSD1 with co-rest complex, okay, when it works with co-rest complex, it demethylates H3K4 methylation mark. So it removes the H3K4 methylation mark. Okay, so it catalyzes this reaction. Okay, so H3K4 methylation mark is an active mark. So removal of active mark, this will lead to the repression of the gene. Okay, this will lead to repression of gene. That is why here LSD1 is kind of acting like a repressor. Okay, so it is removing the active mark. So along with the co-rest complex, along with the PRC2 complex. Okay, when we will learn about long non-coding RNAs, after 2 to 3 lectures, we will learn about long non-coding RNAs. In that lecture, we will see how LSD1 with PRC2 complex removes the H3K4 methylation mark, and how it helps in the epigenetic regulation. Okay, so that is why LSD1 activity is the context dependent. So when it works with androgen receptor, it acts as an activator because it is removing the repressive mark, but when it works with the co-rest complex or PRC2 complex, it is demethylating the H3K4 methylation mark, which is an active mark, and that is why it is working as a repressor. So this was the interesting part about LSD1. So this is the only demethylase that we need to learn in detail. So that's it about histone methylation. So we have covered about in the histone methylation that how histone methylation is done, what are the specific marks, all of those specific marks. What are the characteristics of marks, where in the chromatin those are located, okay, like in the transposable elements, or in the pericentromeric or the telomeric region, or wherever there is transcription site. We have learned all of this in the previous lectures. We also discussed about the enzymes, the HKMTs and histone demethylases. So till now we were only discussing about histone lysine methylation. In next lecture, we will discuss about histone arginine methylation. And with that, we will be done with the histone methylation part. And after we are done with histone methylation, we will move on to the next histone modifications. Like we have only discussed about histone acetylation yet. So with this, we will complete the histone methylation. Then we have to discuss histone phosphorylation, histone ubiquitination, and after the histone modification series, we will continue discussing about other topics like long non-coding RNAs, and then X chromosome inactivation, dosage compensation. All of those parts will be included in this epigenetic series. So I hope you are enjoying this series. So that's it for today. Till then, see you. Bye bye.